Now that we know how DNA can make mRNA, which will travel out of the cell to help create proteins, we're going to see how we go from mRNA to amino acids to polypeptides to proteins, which ultimately determine the traits that our body has and expresses and what makes us us. So the first thing is the mRNA travels out of the nucleus and needs to find a ribosome. Ribosomes are made up of RNA as well. It's actually called rRNA for ribosomal RNA. Now, every 3 nitrogen bases is what we call a codon, and they will code for a specific amino acid. And these amino acids are carried around on tRNA molecules, which you can see one here, and over here, and over here. And each tRNA molecule pairs with a specific amino acid. That's its match. And part of that is determined by this 3-base codon. So what happens is mRNA hangs out in this rRNA like a little hamburger there. The tRNA matches up with the 3 bases in the rRNA. So if the mRNA, if the mRNA has the bases A, U, and G, the tRNA has what we call an anticodon that matches up with those, and we know these hydrogens bonds form, and A has to go with U, U has to go with A, and G has to go with C. And so on the bottom of these tRNAs right here is the anticodon that matches up with the mRNA. Meanwhile, there's the little amino acid that's on the top of it. So it gets pulled in, shifts over, and time after time, these amino acids slowly get joined together. And as you get more than one amino acid, that's called a polypeptide, because amino acids joining together is called a peptide bond. So an amino acid plus an amino acid, this is a peptide bond in the middle. And it keeps going until it's told to stop. And so there's actually a codon that starts things and a codon that stops things, and then all those codons in between that keep adding amino and amino acids until you get your new protein here. So here's an example of that cheat sheet for the codons so that the amino acids know what to do. And it's a little tricky to read, but if you notice, here is the start codon, and this is the first nitrogen base, the second one, and the third one that goes with that start codon. So AUG will be the start codon, and that'll say, hey, you need to start making your amino acids right here. Likewise, once all the amino acids have been put together, and these are all abbreviated here a little bit. So for instance, this is leucine, is the full name of that amino acid. So you might have CUC for leucine, and then that amino acid, the tRNA, is going to match up with that, which is going to be GAG, and there'll be that tRNA thing with the little amino acid on top of it, and that's what will go into the queue. And then maybe the next code in the RNA is CCC, which is the code for proline. So if you have CCC, and once again, this is in the mRNA here, mRNA, and this is the tRNA. The tRNA complementary base strand for CCC would be GGG, and you'd get your tRNA there with that amino acid, and then these start to join together, And we learned earlier that they join together and water is produced. So it's called a condensation reaction. And you get enough of those chains together, and amino acid after amino acid join together to make a polypeptide or protein chain. And so here's kind of the key here. One of the things to notice is that there are 21 amino acids in the body. And if you do a little bit of math here, you can go, okay, if there's 4 possibilities for the first nitrogen base and 4 possibilities for the next one and 4 possibilities for the next one, there's actually 64 possibilities in this entire chart that you have here. Okay, so there's 64 possibilities, but only 21 amino acids. So as you can see, for instance, with serine, you could have UCU, you could have UCC, you could have UCA, or you could have UCG. So multiple combinations will code for the same amino acid. So in summary, you have a DNA strand. Here's the DNA strand in question. This RNA is made through transcription, and then the mRNA will end up linking up with a tRNA that is complementary to this. So you'll have C, A, C, G, U, A, G, A, C, etc., etc. And these are the amino acids that are connected to that tRNA, and then these start to join together through condensation bonds until you get a long polypeptide chain or a protein.